The overall aim of the BalkanPolliS project is to address gap in taxonomic and biodiversity knowledge on insect pollinators in the Mediterranean basin as an essential step toward developing good and effective conservation strategies. So basically, you cannot protect what you don't know or you're not even aware of. And that is why in our research we strive for a comprehensive understanding of the biodiversity around us, especially because we have that joy to live in the Balkans, which is one of the most important biodiversity hotspots in Europe. But this challenge cannot be fully addressed without an integrative approach based on both morphological and molecular data. This is particularly relevant if we consider that one of the major challenges in the biodiversity studies is the presence of cryptic taxa represented by different species that appear to be morphologically identical, but on the other hand, they manifest considerable genetic disparities. And in such cases, there is always a risk of underestimating the true species diversity if we do not analyze molecular variability and base our research solely on traditional morphological characters. So with this in my mind, I can say that my role in the BalkanPolliS project is to shed a light on this hidden cryptic diversity with the attempt to increase the overall taxonomic knowledge by providing an extensive insight into the biodiversity of pollinators in the Mediterranean basin. There is a saying that you don't know what you have until you lose it, and that is exactly what we are trying to prevent through our efforts in the BalkanPolliS project. So in this regard, one of the goals of this project is to share knowledge about pollinators' biodiversity and its value with the aim of raising awareness among local communities about the wealth they literally have in their backyard. So why is this public education so important in terms of biodiversity conservation? Because even though linking our expertise to different regulation or laws gives us a great opportunity to develop good and efficient conservation strategies, we still need this public pressure to ensure a longevity of the adopted measures. So in this way, by supporting local communities to make informed decisions, we increase the responsibility of all actors in the conservation chain. And in my opinion, this is the best and probably the only way to maintain a system sustainable. Although a variability of the mitochondrial CO1 gene has been proved as a powerful tool in identifying and distinguishing a number of insect species, an increasing number of studies report on the limits of the single gene approach. Namely, in cases when only mitochondrial DNA variability is considered, different evolutionary processes such as introgression or incomplete lineage sorting may lead to deep intraspecific splits or haplotype sharing between different species, causing in these cases over or underestimation of true species diversity. And therefore, with the aim of answering such taxonomic questions, additional molecular markers originated from different parts of genome, especially nuclear one, should be introduced into analysis in order to overcome limitations of the single gene approach. But this process is not always so straightforward for non-model organisms such as Hoverflies and wild bees, because of the lack of informative and reliable diagnostic markers that could be used in assessment of management units. But however, in recent years, the development of modern genomic technologies has enabled discovering and genotyping of hundreds or thousands of molecular markers distributed throughout the genome without any a priori information about the reference genome, which is one of the major issues in studies on non-model organisms. So considering that this produced genome-wide data provide insight into multiple loci spread across both nuclear and mitochondrial genome that are under the influence of different evolutionary processes, this kind of data have great potential to finally resolve taxonomic and phylogenetic questions held by this challenging species group of complexes. Due to the complexity of the Mediterranean climate, geology and topography, thousands of geographically isolated populations inhabit islands, peninsulas or mountain ranges where local differentiation has resulted in a great number of endemic species which are particularly sensitive to the environmental changes. But a large part of this species diversity has not been identified so far because of the high level of cryptic diversity, especially within insect pollinators groups such as Hoverflies. And this is particularly relevant if we consider that biodiversity parameters such as species richness and endemism are taken into account in designation of protected and key biodiversity areas. So in this respect, genetic research can significantly contribute to revealing hidden diversity and thus make informed decisions regarding the conservation of insect pollinators in the Mediterranean basin. But also, in addition to defining evolutionary significant units, genetic diversity represents a valuable source of the species' adaptive potential. So, therefore, a selected panel of genomic data can be used to explore signals of local adaptations and therefore enable identification of environmental factors that have a significant impact on pollinator distribution and diversity. And these findings can therefore additionally help to identify taxa and areas that are crucial for conservation and monitoring. In my opinion, every development in this field represents an extraordinary advance in its time, since it always allows us to take a step forward in gaining a deeper understanding of the biodiversity that surrounds us. In our time, this progress is represented by development of various omics approaches that allow us to take a comprehensive insight not only into DNA sequence variability, but also into different mechanisms at the molecular level that are responsible for the functioning of organisms and their interactions with the environment. And this is particularly important with regard to the impact which different human activities and ongoing climate change have on pollinators' diversity. So, essentially, by integrating knowledge obtained from this omics data, we are able to explore the basis of pollinator responses to the various pressures, such as, for example, pesticides or disease, but also to identify adaptive phenotypes and predict range shifts, or, on the other hand, to define species that are particularly vulnerable to environmental change, gaining therefore a valuable insight that can help define long-term management strategies. I believe that every scientist's journey begins early in childhood, and that our family members are our very first mentors who inspire us to interact with the environment and also support our tendencies to explore. Then throughout our education, we meet different professors, scientists who inspire us and whose work we admire. In my cases, this is when I desire to delve deeper into genetics and employ this diversity as a tool to better understand biodiversity. But probably the most valuable lesson I've learned is that you cannot achieve anything alone and without integrating efforts with other scientists. And That is why I can say that the most important inspiration is one that we find every day through the work of our colleagues or team members, because this is the only way we can grow together and maintain scientific progress.